Hello 515 Diploma students. In your next two lab modules, you will be carrying out transcriptomic and proteomic analyses of yeast, that is Saccharomyces cerevisiae, grown with either glucose or galactose as a carbon source. In this short video, I'll show you the culturing and harvesting of yeast cells. Once again, I'm working over a flame as part of sterile technique. The upward airflow caused by the flame prevents particles from falling onto the surrounding workspace. From a single colony of yeast grown on an agar plate, I start 5 milliliter starter cultures in growth medium that has either glucose or galactose as a carbon source. When you're done with the burner, shut it off. Don't leave a flame unattended. The small starter cultures are grown at 30 degrees overnight with shaking. After the small cultures have grown, I use them to inoculate 125 milliliter cultures in growth medium that each have either glucose or galactose as a carbon source. It's a good idea to sterilize the lip of the tube or flask when transferring cultures. I'm using a 4% inoculum to start the larger cultures. These cultures are grown for 3 to 6 hours at 30 degrees with shaking, until the optical density of the culture is between OD 0.8 and 1.2, as read from samples measured by a spectrophotometer at 600 nanometers. I aliquot 10 milliliter volumes of each of these cultures into several falcon tubes. Then I pellet the cells by centrifugation. The cells form a pellet at the bottom of the tube. I remove the supernatant, first by decanting, then by pipetting the last bit of liquid medium. The cell pellets are frozen and stored at -80 degrees Celsius for subsequent RNA or protein extraction.